talk a little bit about organic chemistry. We know organic chemistry is the branch of science dealing with the element carbon and its properties. Most of the compounds found in living things other than the water are organic, carbon-based. About 30% of an organism's dry weight, its dry biomass, is made of carbon. Most of the rest of that mass, that other 70%, is mostly water. Carbon helps to make up four major groups of what we call the organic macromolecules or biomolecules. And those are carbohydrates, lipids, proteins, and nucleic acids. The original source for carbon in pretty much all life is carbon dioxide taken in by plants or algae or cyanobacteria from the air, and then during photosynthesis converted to some type of organic molecule, usually beginning with sugars. Carbon has several important properties that make it ideal for building biological molecules. Number one, it has 4 valence electrons. And this means that it can form 4 covalent bonds. It can bind to 4 different things at the same time. This allows it to form a variety of molecules and, more importantly, a variety of shapes. And since structure determines function, these 4 valence electrons allow carbon to make molecules with lots of different functions. Carbon atoms are small. This means when they form a covalent bond, the shared valence electrons are very near the nucleus. This makes the bonds very, very strong, very, very hard to break, and thus an excellent building material for life. And then another important thing is there's a lot of carbon on Earth. It's very abundant. It's not the most abundant element on Earth, but there's plenty here, and that makes it, again, a good building material for life. Let's talk a little bit about organic molecule structure. Very small, single unit organic molecules are called monomers. This might be like a glucose molecule or an amino acid. But many of the biologically important organic molecules are very large. They're called macromolecules or polymers. These are made up by combining lots of individual monomers together. Usually the monomers are linked together by covalent bonds, very strong ones, and that the molecules or polymers are built and those bonds are formed during what we call dehydration synthesis reactions, or you may also hear them called condensation reactions. During these reactions, the monomers are covalently bonded and water is given off as a waste product. Macromolecules can also be broken apart, like during digestion, for example, through reactions called hydrolysis reactions. These reactions require water as a reactant, and they end up breaking the polymers down into monomers. Here we can see those 2 types of reactions illustrated. So this top reaction, we have 2 small molecules, 2 small monomers. They react together. Energy here is a reactant, and they're linked together to form a larger molecule, a polymer. So monomers make polymers, and notice that water is given off as a waste product. Down on the bottom, we have a large molecule. Water is used as a reactant. In this case, some energy is released during this reaction. The large molecule is broken down into 2 monomers. So that's a hydrolysis reaction. So we're only going to talk about one of those categories of biological macromolecules because it's the one that you're most likely to be asked about on an AP exam. And those are proteins. They are probably the most important category of organic macromolecules. Proteins make up your bone, your muscle, your skin, your hair. Maybe the most important proteins in your body are the ones that serve as chemical signaling molecules or the ones that serve as your body's enzymes. Something that should tell you how important proteins are, the only function of DNA in your body is to code for the structure of proteins. So DNA tells your body which proteins to make and how to make them. Proteins make up more than 50% of an organism's dry weight or biomass. Typically, the names of proteins end in the letters LIN, while the names of enzymes, which are proteins, end in the letters ASE. Proteins are very large macromolecules made of monomers called amino acids. 20 different amino acids make up all the proteins in all of life. Most proteins and enzymes have hundreds, sometimes even thousands of amino acids in their structure. Another thing to know about proteins, not only are they large, they're also polar or charged. So here's some amino acids. So this diagram kind of shows us the basic structure of an amino acid. There's a central carbon atom. Connected to that on one end is what's called a carboxyl group, a COOH. Here this is in pink. It can act as an acid because this hydrogen atom can be released to the environment. On the other side, directly across from the carboxyl group, is an amino group. This side can sometimes act as a base because it can accept a hydrogen ion and it can become NH30+. The alpha carbon is another way to sometimes refer to the central carbon atom in the middle, this one right here. And then the R group is the part that's different in all the 20 different amino acids. So all these top pieces are the same in every amino acid. The R group is the part that differentiates the 20 different kinds of amino acids from each other. And since this part is different, it gives each amino acid some different properties like polarity or non-polarity. So individual amino acids get bonded together by a covalent bond during a condensation or dehydration synthesis reaction. When we're talking about proteins, that bond is called a peptide bond. It's very strong, it's hard to break. In order to create that peptide bond, the amino end of one amino acid, which we see here, is joined to the carboxyl end of the adjoining amino acid. We see that here. So the bond forms between those two. We see that here in green. And again, that's a covalent bond. It's formed by sharing electrons between the carbon from the carboxyl group and the nitrogen from the amino group. When two or more of these amino acids are joined together this way, we can call the structure that results a polypeptide chain. A protein is usually made of several polypeptide chains that are all wrapped together into a large three-dimensional unit. Remember that the function of a protein is usually determined by its shape or its structure, the way those chains wrap together. So we talk about the levels of protein structure on typically 4 different levels. The primary level, or 1' level, this is determined by the sequence of the amino acids. Which amino acids are present and what order are they in? This is the part of a protein structure that's coded for by DNA. A mutation can change the primary structure of a protein, and this can vastly change its overall shape and its function. The secondary structure, or 2' structure, refers to the shape of specific sections of each polypeptide chain. Hydrogen bonding between polar amino acids can cause certain parts of those chains to overlap and coil into 3-dimensional shapes. The proteins have to be folded to work. The polarity or non-polarity of the R groups typically determines how the polypeptides form. For proteins that are built by ribosomes that are located on the rough ER, this folding usually takes place within the rough ER itself. And then those proteins are usually exported from the cell or they get embedded in the cell membrane. Other proteins are constructed by ribosomes that float freely in the cytoplasm. These proteins usually remain in the cell, in the cytoplasm, and work within that cytoplasm. They're folded inside a different kind of protein called a chaperonin. These protective structures help to allow the proteins to fold without being impacted by the nearby water. There's 2 common types of secondary structures that we talk about. They're the alpha helix and the beta pleated sheet. Here we can see the alpha helix, and you can see all these hydrogen bonds that are forming between nearby amino acids. Here we see a beta pleated sheet, and again, notice all the hydrogen bonds that help to hold that sheet appearance in place. Let's also talk about the tertiary structure, or 3', 3rd level. This is the overall shape of a particular polypeptide chain. In addition to the hydrogen bonds, these tertiary structures are stabilized oftentimes by what are called disulfide bridges or disulfide bonds. The amino acid cysteine contains sulfur. If 2 cysteines end up nearby, the sulfurs in those cysteines will form these disulfide bonds or bridges. These bonds are not quite as strong maybe as a normal peptide bond, but they're much stronger than hydrogen bonds. So they help to stabilize the structure of the protein. Lastly, we'll talk about the quaternary or 4' structure. This is when 2 or more polypeptides are wrapped together to form the overall structure of a protein. For example, the protein hemoglobin in your blood, the protein that carries oxygen for you, has 4 polypeptide chains that get woven together to make this distinct 3-dimensional shape. Lastly, let's mention denaturation. So denaturation is when a protein shape is unraveled or unfolded. This causes it not to work. This can be caused by pH changes, either increasing or decreasing the pH, salt concentration changes, either increasing or decreasing the salinity, or increases in temperature. Notice we didn't say decreases in temperature, only increases in temperature. So denaturation usually disrupts either the secondary or tertiary levels of structure because it affects either the hydrogen bonds or the disulfide bonds, which help to stabilize those structures. That's it for Unit 1.